The idea of the lecture today is to, we are back in business of the treating disease with light. So what is the opportunities that we have to improve the efficiency of a certain drug by targeting intracellular organelles? So I made a syllabus today. The lecture is going to be a bit longer. It's actually not 2 lectures, but 1.5. So I will also make it interactive, so I hope we can stand along 1.5 hour, a bit less than that we have. So first I'll speak a little bit about historic perspective of part of the PDT history and how selectivity comes in the beginning of this methodology. And then I will give some background from my lab and for other labs to justify the reasons to do intracellular localization. And then we have for the question of the lecture. So I'll ask the questions and hear opinions. If you pay attention to the background, you probably already quickly answered the question. Let's see how it goes. And then the most part of the lecture, I will discuss data from my lab, of course, compared with others, in which we compare the relationship between localization and different types of regulated cell death mechanisms, and to discuss how to develop new generation photosensitizers to the end. So a bit of historic perspective, looking into selectivity, and also discussing a little bit of companies that were generated, some are still alive, and how they were started and what happened with them. And so this is perhaps, I'm sure it's the most important success in terms of commercial application of photodynamic therapy. So this is application of a drug that was synthesized by, do I have a pointer or something? I think it's here, right? Was synthesized by a scientist, David Oven. He was an organic chemist. He worked a lot in the initial development of PDT in very important research centers in the US, and then moved to Canada. And then he had many different compounds, and he found the company KLT Incorporated for a drug. It's called vetoporphyrin, which is this one, to treat age-related macular degeneration. So there is an increase in the vasculature here in this region, and person becomes blind if nothing is done. At that time, the only treatment that was used against this type of disease was using infrared light to treat this, and then half of the people that were treated got blind for the whole life. The ones that were not treated also got blind, so PDT was really a good success. Around 70% of the people could recover from this blindness, age-related macular degeneration disease. And this made a lot of money. This company made an agreement with Novartis, and they sell it everywhere. So ophthalmologists are real aware of the PDT concept. They made, in 2002, $350 million US per year. However, soon after this, the same company developed a drug that is an antibody that also targets the vasculature endothelial growth factor. So if you inject this antibody in the eyes, they will target this endothelial factor, and there is a smaller growth of this vasculature. So for some time, the person recovers the vision. And this has to be used many times, and it costs a hell lot of money for this. But this company decided not to go anymore with vetoporphyrin. This company, KLT, decided to go for ophthalmology in the middle of this big boost of money, and then now it's basically sold all the patents, no longer something that's really investing or doing any new developments. So that's an interesting case we should consider. And in my view, many, or perhaps all, PDT companies started with person. So I guess we depend on you. So you are the new entrepreneurs. You have to think of how to do it, and be careful with the big pharmaceuticals companies because they don't like to sell light. So How do I change this? The slides. This is not working. No, I can do it here. So we did in the lab some research related with this chlorine compound. Instead of this structure that I mentioned from vertoporphyrin, a student made a smaller, different structure, and this group is a lot more rigid than the other one. And so these molecules does not aggregate. One of the major problems of vertoporphyrin is that they need to do a very difficult separation, because the other derivative that this group enter here basically aggregate and avoid completely the effect of this molecule. So they had to separate. And in this drug that we developed, does not aggregate. I mentioned to you some data yesterday, so it's a L shape. And there is an ophthalmologist, his name is Gustavo Salomon. He tried this with rabbits. So the molecule was injected as a liposome suspension. You see this is the absorption of the free molecule, the liposome suspension, and put red light in the eyes of the rabbit and saw that this is the exams that are usually done in retina studies. And so they saw there was blocking of the vasculature in this region that's supposed to be. And when you do for histopathology, this is the vasculature that has to be destroyed for treating AMD. And you see this was done, but the retina and the sclera, the other organs that has to be kept, were not damaged. So another thing I want to discuss in terms of PDT history is this compound, aminolevulinic acid, which is a precursor of porphyrin. So we all need to make porphyrins to make heme groups, and the porphyrins are made in the mitochondria. This part, so ALA enters here, and there is porphyrin coming out, okay? And porphyrin, if it doesn't do chelation with iron, it becomes a very important photosensitizer. So this really make a revolution in terms of PDT, because you didn't have to consider putting the photosensitizer itself. You put a prodrug, something that will turn into the drug inside the cell. So, so the mechanism of cell death that this cause is apoptosis. I will discuss a little bit more what the meaning of this in the following slides, but just to give you some information. And so when this started, so the year of ALA that really the community got to get to know this is 1992. Photochemists were not the people discovering that ALA was used to make porphyrin, and that if you remove iron or if you chelate iron, the cell would accumulate a lot of porphyrin. So this was previous research, but the photochemists were the ones that realized they could use this concept to make PDT. So these several different groups around the world, okay, they published sequentially this. But the group that really made this into a technology was the group of Norway. There was this guy, Moen, and these people start to develop for PDT in the early starts, and they were also competing here. But in the end, they found in 1993, so just after the publications, they found this company called Photocure. It was basically for using ALA. And ALA is still a success. So you see, it still makes $60 million a year. Now DUSA is the company that's selling it. And Metivix was also developed by Photocure, which is a derivative of ALA. There is a methyl group there, so it's more lipophilic, enters better to the cell, it's more efficient. Now Galderma is selling this, and it's making 50 million euros per year. So another example in which a scientist, with help of company, developed the technology. Okay, and this is still alive. Okay, here in Brazil, the group here in São Carlos has everything to transform this into also Brazilian technology, but Anvisa is not helping so much. So you can talk to Christine about this if you want. But anyhow, think of this scientist. He used to be a porphyrin chemist. I mean, he liked to study in the cells how much molecules enter the cell, what is the concentration of compounds inside the cell, how this really explains the activity of this specific compound, the amount it enters the cell. Think of this guy looking at the data in which he makes the molecule inside mitochondria, normally, but just treating with a molecule that is not a porphyrin. So this was a big revolution, and has to do with targeting, what we are discussing in this lecture, making this photosensitizer target specific organelle. So this is perhaps the first example in which this is a very, very clearly example of targeting PDT. So this was a historic introduction, and you can ask me questions and stop anytime, please. Do so. I would like to. So let's go for a bit of justification. Why do we need to think of a molecule that is more efficient? Why do we need to consider developing more efficient photosensitizers? What's the reason for that? In my view, the reason is the following. PDT has the potential to really transform many disease treatments. We saw data here in this, several lectures in this workshop, in this advanced school, and there are many others that are published already. And the reason why there is this potential is that there is this photonic revolution that we are discussing here, photonics, but the cost of light is so much cheaper today compared to 20, 30 years ago. So this is amazing, the transformation that occurred in this field. And also, the device that can be used to put light anywhere we want. So this is really ready to be used. But the photosensitizers, they are based on the same principles we developed 40 or many years ago. The Egyptians used this same concept. I mean, how can we make it work better? And why do we need it to work better? The reason is, okay, if you have a lot of light and a lot of dye, a lot of compounds inside the cell, no cell will survive. Mark was telling you some of the reasons why this happens in antimicrobial PDT, but you can kill any cell, actually. So that's something else we should consider when you do studies in the lab, in vitro studies. You go there and you kill your cell culture. What does it mean? Nothing. Because if you raise the pH or if you lower the pH, that would kill the cell also. So you have to find out good questions when you do studies that are in mimetic system, experimental system. But here, okay, but so depends on where this tumor is, or depends on where the infection is. You actually cannot have a lot of light, or you cannot have a lot of dyes. So that's the reason to have survival of this disease tissue or bacteria or cancer cell, and this will come to the disease will not be cured. So if we make a molecule, kill a cell with a very small amount of light and a very low concentration, we are improving the efficiency of this molecule. So that's why I think we should consider ways to improve the efficiency. And the way we think it's important is to learn more about the mechanism. If you learn more details, you'll be able to design better drugs. That's the main idea. So different experimental possibilities. So you can treat people. And you have to treat the disease, and it's fine, case reports, clinical trials. You can treat tissues. So you were talking about 2 lectures, very nice lectures from Avagdor yesterday or Zhang today. Tissue targeting or vasculature shutdown. So this is really important. So how you make the drug concentrate, focusing on this disease tissue. I think it's one of the most important aspects. But it's not the one that I deal with in my lab. So I leave this to other people like Zhang and Victor to discuss this. So we start to bother here, we start to worry in my lab in this area. How can we make a molecule that is more efficient in killing a cell? So if it's there, is it as efficient? How we can understand the efficiency compared with the structure of the molecule? and we always have to go down certain levels. So yesterday we discussed a lot about the membranes. So what are the specific reactions between the photosensitizer and the membranes, how we can maximize this? And we can do the same with organelles. And that's the issue of the lecture today. When we damage organelles, how the cell respond? And how we can use this information or this response to make the molecule become more efficient. So again, remember of yesterday, we are discussing photosensitized oxidations. You excite the light, you form a species that live long enough or is bound to a target. There is electron transfer reactions. On this, you end up damaging the biomolecule, the protein, DNA, or membrane. Sometimes this causes photobleaching. There is other mechanisms to form reactive oxygen species. You have suppression from the triplet forming single oxygen. You suppress the radical, anion radical, or the semi-reduced radical formed by forming anion radical superoxide. So many possibilities here, always using light to do reaction, okay? Yeah, I also showed this slide yesterday, so what people have done in the last years, many years, going from porphyrin chlorine to bacterial chlorine to make light absorption in the red, maximizing reactive oxygen species. And then we ask again the question, is this an important parameter? And today I'm going to share with you one data that I didn't show yesterday, which I think is perhaps the most clear example that really doesn't really matter how much you make of a radical or a reaction. What most important is where this reaction is happening within the cell. So we are ready to compare here two molecules that are positively charged. One is methylene blue, and does both type 1 and type 2 reaction, very efficient, 0.5 single oxygen content yield if there is oxygen. So it makes, in a certain condition that I'm showing the data afterwards, 20,000 single oxygen molecule per second per femtoliter, okay? Very efficient. This one is crystal violet. You see these rings here. They are kind of rotating. So as soon as they get to the excited state, they go back by releasing heat. So they're very poor photosensitizers. But they do a little bit. So the yield is a lot lower than 1%. They do both. They can make some single oxygen. They can make radical. They can react. They are redox active. But they do a very small amount of reaction, okay? Both are positively charged. And they have lipophilicity that's similar. Not the same, but it's similar. So if you look at the data, so this is data of toxicity in the dark and under illumination. You see that both of them, when you put light, they become a lot more toxic. This is culture of HeLa cell. We can measure amount of incorporation. Both of these dyes incorporate in the cell in a certain limit from 70 to 80%. Because we know how much these dyes absorb and how much they are inside the cell, we can estimate how much light they are absorbing. And if we know how much they are killing, we can do like a relative toxicity that considers the amount of photons absorbed. And when you look at the data, this dye that is very poor, does not make much photochemistry, is actually at low concentrations more efficient than the other one. See, it kills better than this one. If you start to increase the concentration of both, they become similar at a certain concentration. Okay? But it's an interesting case to study why are they similar, or at least low concentration. This is even better. Okay? And so we went to study more details of what happens inside the cells. So both of them, I think now start to work. Both of them are killing the same arbitrary units here. This is cell death. We measure single oxygen quantum efficiency within the cell. Okay? We put cells in the spectrometer. We have emission spectrum in both cases, but this one is very small. Okay? Very hard to see this. It's possible to see, but it's very small. And this one is a lot of single oxygen inside the cell. And this is 50 times more than that. So it makes a lot more single oxygen, but it's still killing the same. So we thought maybe there are other radical species that are involved. So we compare now oxidation of a probe, like chlorofluorescein. When this probe gets oxidized, emits light, and then methylene blue still oxidizes a lot more of this probe compared with crystal violet. So what else could we see there now? So now I'm going to stop. You are the scientist. What else should we study? So I'm telling you that these molecules are very different in terms of photochemistry. They kill the same. And this is not only in the test tube, this is within the cell. Yes?
May we see the localization of both the compounds? Because depending on what organelles they are targeting, it will change.
Perfect. So you want to work with me? Yeah, I have a postdoc position, so... Exactly, that's what Carla did. Carla Colum, she was working with me at that time. So she did a lot of co-localization protocols. So this is crystal violet emission, this green one. She used rhodamine 1 through 3 that targets mitochondria. She puts one over the other, 100% co-localization. So all this dye, it's not a lot of dye, it's a small concentration, but it's all inside mitochondria, okay? It's very hard to find this type of co-localization. Even if the dye is positively charged, it's not easy to find 100%. But this guy goes 100% to mitochondria. It doesn't go to lysosome. You do lysotracker, nothing. Body P, it doesn't co-localize also with reticulum. It does not go to the nucleus. What happens when we do it with methylene blue? When we're doing the conditions that co-localization studies should be performed, and this is very important, should be a very low concentration of the dyes, otherwise the dye distributes to all sites. So you don't see any emission from methylene blue, actually. It's basically nothing coming out from the cell. So the dye is suppressed somehow, and I'll explain to you later on what happens of the suppression. Actually, it's here. We have data afterwards to show that this dye gets reduced inside mitochondria. So it does localize there, but gets reduced. So there is no absorption, no emission, no photochemistry. But if you put a rhodamine, the dye that marks mitochondria, and put methylene blue over it, you see that it's redistribution of rhodamine. Rhodamine was in mitochondria and goes out because this dye is going there. So this is a competitive assay, so the dye is there, but it's suppressed. And also if you go for a higher concentration, which is not really the best way to do co-localization study, but what happened is that co-localized really well with lysosomes, okay? And so that's the end of work of Carla. She published a nice paper, well cited, but what is the concept here? Really, where this dye is located is a lot more important than the amount of whatever ROS or single oxygen is generated. Mitochondrial localization is very important, so we should look into specificity. So what happened with these dyes when they are in mitochondria? So for some time we studied photochemistry of them inside mitochondria. And so look at, this is just an example, we have others in the lab, but this is becoming very clear. This is mitochondria. I hope everyone knows what I mean by mitochondria. Does anyone not know what mitochondria is? Please raise your hands, I will explain. So this is the eukaryotic cell power plant. It makes ATP, and makes ATP by making a proton gradient. So this side of the membrane becomes negatively charged. That's the reason why mitochondria attracts positively charged dyes. But if you add a compound that's called CCCP or any compound that will allow protons to permeate the membrane, you remove this negative charge. And so look what happened with this guy, methylene blue. If it is under mitochondria that is breathing, that is working, generating ATP, you see that's the absorption spectra. It's a clear absorption spectra of a monomer of this species. However, if you make mitochondria breathe, a lot of dye goes inside, and you see the dimer spectra. So it changes the composition based on the mitochondria if it's active or not. And this makes a lot of difference in terms of single oxygen. when the mitochondria is not working, it generates a lot of single oxygen inside the cell. But if it's working, there is this dimer species. Dimers do not make single oxygen, so it does not make single oxygen inside the mitochondria. And someone yesterday asked me about this spectral features of methylene blue, so I decided to show it here. So methylene blue makes triplet species, and we can see here in this triplet absorption spectra. So what happens is we make the triplet, we shine another light, and we see the absorption of this transient species. What is the spectra of this? So this is the clear absorption spectra of triplet methylene blue. However, when you are in an energized mitochondria, you're not like this, you're like this, and the spectra is very different. So in the beginning, few nanoseconds after the laser pulse, you do have here triplet, but this becomes very short after the pulse, semi-reduced radical that you see the absorption here at around 400 nanometers. And then if you wait some time, because this radical also reacts with oxygen, if you wait for some time, you start to see another guy here, another molecule, which is the oxidized species of methylene blue, and this one here. And then what we propose is that methylene blue has this monomer-dimer equilibrium. When it's in dimer, and this happens when a mitochondria is energized, this molecule goes to the excited state, it has an internal intermolecular electron transfer, forms these two radicals, and then doesn't form single oxygen and doesn't react. So that's why this dye is so, the efficiency of this dye at low concentration is so small, okay, because of all these reactions happening there inside mitochondria. And what is the consequence of reacting species damaging the membrane by mechanisms that we discussed yesterday inside mitochondria? The consequence is the following. You release a molecule that's called cytochrome C, which is here in the internal membrane of mitochondria, is responsible for transferring electron from complex 3 to complex 4, and also if it gets released from mitochondria, it becomes apoptosis. I mentioned to you a bit more what apoptosis means. And so at this time, we want to know exactly the oxidation of the protein or oxidation of the membrane is responsible for releasing it from to the cytosol and to cause initiation of apoptosis. So I just discussed one data in this. We have several papers, but just one data that I want to discuss with you is that in the end, what we prove with technique that we just heard about sulfospasmol resonance, we measured the interaction of this cytochrome C with the membrane, and we observed that if the membrane has this hydroperoxide that are formed by single oxygen, then the protein gets released, the binding constant goes from this type to this type. So there is the Kd here, the dissociation constant when there is hydroperoxide increases many times, manifold. So that's why cytochrome C gets released from mitochondria, because you form these hydroperoxides there. And a previous data I'm not showing here is that if you oxidize cytochrome C itself, so if the protein gets oxidized, it binds more strongly to the membrane. But even if it gets released, because if mitochondria is destroyed and gets released to the cytosol, if the cytochrome C is oxidized, it's not able to induce apoptosis. Okay, so we claim that the main mechanism of apoptosis induction is oxidation of these lipids here. And we prove that hydroperoxide is able to release cytochrome C. So now comes the question. So the question today is why intracellular localization is so important in terms of defining the efficiency of a PDT photosensitizer. Yes?
It's important because it determines the type of cell death mechanisms that will be activated.
Okay, I can hire you for a postdoc also. That we talk about later. He's yours, so after. So yes, so you guys are paying a lot of attention. I like it. So I was going to give you some hints, but I'll go, and then I'll go back. So yes, the thing is, where the damage is done inside the cell defines the mechanism of cell death, okay? This is very important. And here also another important thing that I will discuss later on. I'm talking about regulated mechanism, not program mechanism. And then I hope we can discuss a bit about this in the near future. But before we go there, I want to mention a few people that came to this field a lot before me, and they did very important contributions. So if you want to learn more about this issue, please consult data from David Castle and Nancy Oleinik. They are colleagues, mainly David Castle. Nancy also did some work, but mainly David Castle. So lots of papers from the decade of 90s, mitochondrial photodamage, apoptosis, subcellular localization. So actually, my research was a lot influenced by the results of these guys, and also Prastina Agostinis from London, from Leuven. Okay. So let's talk about regulated mechanism of cell death. So I'm spending perhaps 5 minutes on this. It's important because that's what I'm aiming to discuss with photochemistry afterwards. So necrosis, and now we better define necrosis as non-regulated necrosis, because there are also another branch of cell death mechanism that's called regulated necrosis. There are many, okay? So what happens when non-regulated necrosis? This is basically a condition in which the cell loses capacity very quickly to keep alive. When this happens, if there is a damage in the cytoplasmic membrane, so if this damage cannot be repaired, and if we are talking about making a hole with PDT, we cannot repair this. So this leads to necrosis because cells lose homeostasis very quickly. Also, if you damage enough of the mitochondria that it cannot produce ATP, you cannot go through any regulated mechanism of cell death. Why? Who can tell me the reason for that? Why a very strong damage in mitochondria? It can be in other organelles also, but why the cell cannot anymore engage in regulated cell death mechanism? Yes?
Is it just because it takes energy to actually make sure that it's a regulated process and to do all the blebbing and
Very
stuff?
good. Yeah, I can offer a postdoc also if you want. Well, yes, the mitochondria produce ATP. And for this regulated mechanism of cell death to happen, it uses a lot of energy. So if there is no ATP, there is no way. And many times, the cell starts to do some of these regulated cell death mechanisms, but cannot finish because there is no enough ATP. And then the cell evolves to an uncontrolled mechanism of cell death, okay? So what necrosis, you have a disordered degradation of biologic structure of all the cells. There is also regulated necrosis. Okay, this is a kind of new field. What that means? That means that there are enzymes that are activated when the cell senses that there is a problem. It decides for some reason that it's supposed to die. So there is enzymes that are activated to cause the specific degradation, controlled degradation, okay? And so this is, if you want literature on regulated necrosis, I cite this very nice review that came out some time ago. What is the most famous mechanism of regulated cell death? Apoptosis. And also this was called programmed cell death. And why is it programmed cell death? Because this mechanism is working to make a tissue or body or organism survive, to live well. Why? Because it is programmed in the DNA of the cells to have a mechanism of killing itself. Okay, it's not all the mechanisms that we will discuss today that is in this case. And this was a long discussions for decades, and I'll mention a little bit about this to you. So this, the term apoptosis is very old. It started in 1972, this term was coined. Okay, and all the machinery, all the biochemistry, the genetics involved on apoptosis was on the decade of, in the 90s. And what happened, let's suppose when the fetus is development, before you have hands, before the fetus has hands, it has a mass of cells here. And why we find hands here? Because these cells that were in the middle here, they got killed by themselves, okay? They were killed by apoptosis. Another example, we have white blood cells, lymphocytes. They're activated in the inflammation response. But there are many cells that are activated, and only one cell type will multiply to make antibodies. And the rest, thousands, they will get killed by apoptosis. So it's a programmed mechanism of cell death. So what happens is the cytoplasmic membrane integrity is kept, and the ATP level has to be to a certain level. There is a controlled disintegration of DNA, cytoskeleton, and so on. So it takes time also. So if you are in cell culture and you want to see apoptosis response, there are many, of course, there are many cell signaling mechanisms that occur few hours after the start of the damage. But if you want to see signs of apoptosis, actually you should look for 1 day after you do the damage. Autophagy. Okay, so autophagy was also, has a nice history, 2 Nobel Prize. So you have it 2016, this Japanese Yoshimuro. Christian de Duve, a lot before, was the one discovering lysosome. When he was writing paper, he was also using this term autophagy. So why this new Nobel Prize on autophagy? Because autophagy become a very important target for medicine. The tumors, they are messier than the normal cells. And they only survive because they have a very strong mechanism of degrading and recycling their components. So if you somehow use some chemotherapy to mess up with autophagy, you are in the leading process of developing an anti-tumor drug. So autophagy, it takes time, more time. So you should look for it 2 hours afterwards. So what is this really the process here? What we are talking about is that a mechanism in which the cell digests itself. So if you take any cell in your liver, for instance, you cultivate liver cells, and you feed the cells with all it needs. Autophagy, you cannot really see activation of autophagy. It's blocked, it's silenced. However, let's suppose you give medium. That is the small amount of things for survival, but it lacks a lot of nutrients. So it is a nutrition deficiency culture. And then how the cell survives? By activating autophagy. So it's a survival mechanism. During decades, there was a fight in the community if autophagy is a survival or if it's a cell-killing mechanism, because sometimes you see cell getting killed, accumulating a lot of autophagic vacuoles with lysosome enzymes and all the digestion process. And there was a discussion, is this a programmed mechanism of cell death? Is this programmed to happen? Or is this a survivor didn't work? So this was many years of discussion. There was literature of very important people discussing pro-survivor, pro-death autophagy after the Nobel Prize. I mean, this was clear out because this is a fundamental process of degrading recycled materials. So this is a survival mechanism. And how it gets to be used to kill the cell? You have to block autophagy. That's how you get cell killing. So, and this takes time for the cell to die. So you should look for 2 days, 3 days after damage to start to see signs of autophagy. So you see a lot of accumulation of these acidic vacuoles that Yoshimura is holding there. And that cytoskeleton is kept, ATP level is kept, but you see the cell accumulating a lot of vacuoles, okay? Now, this field is very active. All important scientists, they want to have a new term. So, you look in the literature, there are many, many, many names now. Now, ferroptose, oxoptose, part of each of anyone wants to publish a cell paper and invent a new cell death mechanism. So this becomes boring for me now. I don't go for that. There are many terms, details of ferroptosis. So there is iron is involved in the process of oxidation of the membrane of lysosomes, and then it's called ferroptosis. But I mean, for me, it's a detail. But I mean, I'm not here to discuss all this, but there are many names, okay? This is just one list of them. There are more. Okay, so what do we want to do in the lab? We want to damage certain parts of the cell so that we can control where the cell goes. If it survives, it goes senescence. If it kills by autophagy, if it kills by apoptosis, or if it goes for necrosis, okay? This is the part of the lab, that's what we do. And how do we control this? By controlling where these photosensitizers are located inside the cell. So I'll give you one example of each case. So this is work done in collaboration with my colleague Letizia Labriola and Anselli. This was published a year ago. You see, they are specialists in breast cancer. And there are three modalities of cells here, the breast cells that are normal, this is cancer cell, and this is very metastatic cancer cell. And when they compare the efficiency, and this is just methylene blue photochemistry, you see this is cell death percentage, okay, with 2 and 20 micromolar. What you see here, especially at 2 micromolar, 1 hour, 3 hours, 24 hours, that this cell here gets killed a lot easier than the other two. Okay, look at this. For instance, 3 hours afterwards, 2 micromolar methylene blue, 70% is killed. And the other two, the breast cancer, the normal one, and the one that is cancer, the breast cell, the cancer cell that are not metastatic, they do not get killed as much. So we measure the amount of single chain. In each case is basically the same. So it's not amount of dye that's internalized is basically the same also. The amount of single chain is basically the same. What is the difference? The difference is that this metastatic cell has a very small pool of reduced glutathione. Glutathione is a very important reducing species to keep this redox balance in the cell. And this metastatic cell has a lot less. But the mechanism of cell death here is necrosis, or a mix of necrosis and regulated necrosis. So let's give some examples of autophagy and apoptosis. So again, I will give the credit to my previous student, Tayana, she's there. So if you want to meet her, it's her. That's a PhD dissertation. So she was comparing these two molecules. I'll mention a little bit about them. So this is positively charged. You see both are amphiphilic. They enter well the cell. This is positively charged, so you already know that you target mitochondria. It does. I'll give you some data on that. But leaves lysosome free of damage. And this one is negatively charged. It goes to lysosome and keeps mitochondria free of damage. Okay. And so the reason why it goes to lysosome is that lysosome keeps the inverted electrochemical potential. It needs acidic environment inside to do the hydrolysis. So usually interacts and attracts negatively charged photosensitizer. And there are other types of photosensitizer that also target lysosomes, the ones that do not have a charge and get charged at lower pH, so they also get stuck here in lysosomes. So there are many ways to target lysosomes. So the photophysical properties of these two molecules are basically the same. I'll go very quickly. Tayana was able to study the interaction and damage in the membrane. She actually observed that the positively charged dye is a bit more efficient in terms of membrane damage. Look at the data here for erythrocyte damage. The positively charged dye kills the cell very quickly, and this one takes longer. Other membranes is not as different, but always this guy, this positively charged, is more efficient in terms of damaging the membrane. But then she went to study efficiency of cell death, again with HeLa, and let's look at survival curves. And this is one of them here is, this is proliferation cells, and this is MTT, and they basically are the same response. So you see that they are at dark, they do not respond. We are talking about low concentration, nanomolar concentration. But if you irradiate, both of them kill the cells. But look at the IC50 here. It's at around 20, and this is around 100, okay? And basically the same data here. So if you consider also that this molecule is a bit more incorporated in the cell, negatively charged is 5 times more efficient than the positively charged. And interesting, the positively charged dye is the one that targets mitochondria. Where is the data? Here is the data. So here is the emission. This is the staining of the nuclei by DAPI. This is mitotracker red. This dye stains mitochondria that are alive, stains mitochondria that are breathing. Do not stain mitochondria that is dead, okay? So you see here the control mitochondria is staining. However, if you treat this with positively charged dye and put light, all the mitochondria are dead. This is expected. Okay? We put a positively charged dye in the cell, it goes inside mitochondria, it does all these reactions we discussed yesterday, and mitochondria is dead. So you don't see any red staining by this. However, this other one, negatively charged, do not go to mitochondria, keeps mitochondria alive. I'll go fast, because there are other things we did, but it's the same response. The negatively charged dye, it goes only to lysosome. So you do the control, you put lysotrackers, the same idea here. It stains lysosomes that are intact, and you see it here. The cells treated by positively charged dye and irradiated do not damage lysosomes, but this one damages a lot lysosome. We also do catepsin, which is an enzyme that is in the lysosome and goes out, and we have the same result. So what is the consequence of damaging mitochondria and lysosome? In this case, we know we are only damaging mitochondria. So this is the control, stained with acridine orange, so you can see the nuclei, the lysosomes, everything. And if you treat, this is 3 hours after damage, and there is no incorporation of propydium iodide, which is a dye that enters cells that are dead by damaging the cytoplasmic membrane. The cells that were treated by the positively charged dye, they show a lot of propydium iodide incorporation, so necrosis. And this one, these cells treated by this dye is alive. Nothing, very similar to the control. The only thing you can see is that the staining of the lysosome is a bit weaker than here, suggesting there may be something with the lysosome. But 3 hours after damage, nothing you can say about the cells treated by this dye, okay? But this one, with mitochondrial damage, they suffer, okay? And we also measure other processes that are involved in apoptosis. Annexin is an antibody that recognizes a lipid that goes to the external cytoplasmic membrane when the cell enters apoptosis, and Tayana measured this and saw that this is a mixture of necrosis and apoptosis. It's starting by apoptosis, and then many cells die by necrosis. But these ones are okay. Look what happened afterwards. Where is the data now? Okay, if we look 48 hours after damage, so you see the control is here, it's fine. These cells that were damaged by positively charged dye, some of them were dead, some of them were alive, and they continue to multiply, and they're fine. 2 days afterwards, they continue to multiply. However, these ones, 2 days afterwards, they start to die accumulating a lot of acidic vacuum. You see the red here, okay? And we were able to define in the lab something called the autophagy arbitrary units. I will explain a bit later. And you see this increase a lot this time. And we also able to measure this protein LC32, which is a protein that's involved in the start of the autophagy process. And you see there's accumulation of this dye. And a lot of this is accumulating, showing that there is cell death with autophagy, there is blockage of autophagy. And so, let's go back a little bit. So why is this molecule negatively charged more efficient than this one? If you're comparing membrane systems, I mentioned to you, this is more efficient. Amount of redox or oxygen, single oxygen, whatever, the same. But this one destroys membrane more efficient. However, when you do the cell study, this is more efficient 5 times. Why is it? Who can explain to me? Yes?
First of all, I think it has to do with the colocalization, the fact that it can activate 2 different modes of cell death. And I don't know if you're also going to speak about it later. I also know, like, if it's localized in other organelles, it can express on the surface the DAM protein, death-associated molecular protein, which such as ECHOC protein 70 and 90. And this can also promote necrosis and immune cell death, I mean, in vivo system.
Yeah,
so.
but I agree with you, but I want to see this one. I mean, it has to do with localization, but that we all know. Why? Why is this more efficient? So I'll ask Tayana that. Tayana will have to help me here. Explain. I just want to. So the work is yours.
Okay, because the autophagy is blocked, so the cells die more.
So
Basically,
what?
it's because of the autophagy.
Exactly. So what Tayana shows that if you actually targeting lysosome may be a better way to kill the cell efficiently than targeting mitochondria. Okay, because when you target lysosome, you induce this delayed cell death mechanism that's involved with autophagy blockage. Okay. So what happens then if we damage both? If we damage mitochondria and lysosome, and then we compare 2 dyes, I like methylene blue as a very good example for me. I have been studying it for many times, but comparing with other dyes, we learn a lot of important features here. So I mentioned to you, methylene blue is inactive in mitochondria. I show you the data why. Okay, so at low concentration, this dye is there, but it's not active. However, if we increase the concentration, it damages basically lysosome. And this dye, I'll show you the data, dimethylmethylene blue. It is more lipophilic, but that's not the main reason. For some reason that I'm not so sure why, it does not get reduced inside mitochondria. So this gets reduced by a hydrogenation reaction. This one does not. So probably the enzyme doesn't fit well, I don't know exactly, but it's active in mitochondria and in lysosome. And so, does it damage, when we treat this dye with mitochondria with this dye, we can see that it damages a lot the DNA of mitochondria. So this is mitochondrial DNA. We can do PCR here to detect long piece of mitochondrial DNA. And you see that when we treat these cells with dimethylmethylene blue and irradiate, we damage a lot mitochondrial DNA. Nothing happens with nuclei. We measure the potential of electrochemical potential from mitochondria. This one damaged, this one does not, as expected. When we do, I think there is one more, yes, this. So when we do survival curves, you see that the IC50 of dimethylmethylene blue is 10 nanomolar. So you treat 10 to the C5 cells with 10 nanomolar of this dye, and it kills half of this population. This one, you need 2 micromolar, so 200 times. And besides that, what you see here is that when this is in a condition of IC50, 2 days after damage, there's a lot of accumulation of acidic vacuoles here, nothing there. So, and we can measure things here, biochemistry, and we can see that there is LC3, 2P52 accumulation, and LAMP22A, which is lysosome organelle. So there is autophagy blockage, and we also have this data here showing this is the control, and this is just after damage with dimethylmethylene blue. You see that there is no staining of the lysosome, because the lysosome was severely damaged here. There also, a little bit, but not so much. And so what we propose in this work is that if you damage both mitochondria and lysosome, cell starts autophagy. In this case, it's called mitophagy because it's an autophagy mechanism to save mitochondria, to digest mitochondria. But then, because the lysosome is also damaged, it does not progress. So it's a very efficient way to kill the cell. Do very small damage in mitochondria and lysosome. So how do we prove this? Methylene blue is very good to cause lysosome damage, but not so much mitochondrial damage, at least at low concentration. So perhaps what we propose is that we do an external damage, do both also, and then see what happens. And the hypothesis was right. We used 2 independent chemical agents that damage mitochondria. One is hydrogen peroxide at low concentration, activated mitophagy. We had this data from the literature. We proved this was happening. Rotenone was an inhibitor of electron train reaction, so it's also damaging mitochondria. But when we do both rotenone or hydrogen peroxide and then methylene blue, you end up seeing this type of cell death that is autophagy-related cell death. Okay? So then we prove this concept. So I think you guys already know the answer. Why should we care about intracellular localization? Because basically defines the way the cell dies. Okay? And if we want to kill cells more efficiently, that's the way we should progress. So I should remind you that all cells will die by necrosis, depends on the level of damage that you do. If you do a very strong damage, you put a lot of dye and light for many times, all the cells, they will burn out, okay? That doesn't say anything about the efficiency of the dye, because it happens independent on the cell or the dye. You find a condition that you kill all the cells by a lot of damage. What we should look into a condition that we activated this regulated mechanism of death in order to probe a certain condition in which we will be in a tumor or in a site of infection that there are not so much light and not so much dye, okay? So what we think are the paradigms for development of new photosensitizers. And Professor Zhang gave a wonderful lecture of how to deal with multifunctional. I will mention a few examples of my group. But before that, we published this review that we discuss all the issues that I discussed in this lecture. So I recommend if you want to go into more details of this. So for us, in terms of cell death, we should look into localization first, and also robust photochemistry. Do both, type 1 and type 2. So a few examples in my lab. This is a collaboration with Professor Turro from Ohio State University. She synthesized ruthenium derivatives, for many things. And then she made a series for us to test the hypothesis that if the molecule makes two things, single oxygen, and also it's a kind of a chelation mechanism that's for-induced because these ligands are released by light and they bind to protein, membrane, or DNA, whatever they find. And so how we compare these three molecules, one only makes single oxygen, which is this. This one only makes chelation, and this one makes both. Chelates and makes single oxygen. And so this was done by Cristiane Pavani. She's a professor now. So the thing is, this molecule that do both, this is the efficiency regulated by dark and light efficiency, the ratio. So you see this is 1,000, so there is no unit here, compared to 280 for the dye that only makes single oxygen, compared to 6.4 for the one that do only chelation. So the synergistic effect of having both is an important factor. So I have many things I will go quickly. I will not discuss with you because there is no time. I will go more into this topic of how can we use nanomaterials to control the mechanism of cell death. So talking about apoptosis. So this is a work we did in collaboration with a professor from chemistry called Koichi Araki, and we supervised Diana Eda. She was using this molecule, it's a porphyrin, but it's not very soluble. If she wants to treat the cells with this porphyrin, she has to make a suspension in DMSO water and treat the cell. And then when she treats the cell, this is the red fluorescence of the molecule. She goes to the cytoplasmic membrane. You shine light. You kill all the cells by necrosis. How do we know? Incorporation of propidium iodine. However, if she treats the cell not with the suspension in DMSO, but using this nanocapsule, it's called made of biological materials, conservated nanocapsules, and she then together in the cell, and you see here we can stain the nanocapsule with blue here, and the red fluorescence. You see it gets released inside the cell, and then inside the cell you see it has a positively charged, and then it was very likely to go to mitochondria, and it goes. And if you shine light, we have now very clear feature of apoptosis cell death. So we see a caspase 3 release, we see cytochrome C release, and this is an annexin response. Annexin, again, is a marker of apoptosis because there is a lipid that is expressed in the external membrane when there is apoptosis. Very quickly here also, I just want to give you a bit more of the rest of the dissertation of Diana, because she reached this point in which the molecule, this is more efficient than that. But then she spent some time in New Jersey, in this center of biomaterials with Joachim Kohn, and she devised a way to incorporate these two molecules into something called the tyrosphere. It's a polymer, a tri-block polymer that is made of ethylene glycol and oligomers of tyrosine, octioesters. And see, this is an example of the polymer. It can fit 80 of these molecules in 70 of that. This is still unpublished. We are working, Joachim is sitting in the paper now, but we have to talk to him, Diana. Anyhow, less molecules incorporated. When you deliver it as a suspension in the nanoparticle, you actually decrease a lot the amount of incorporation of the porphyrin. So less gets incorporated. However, if you measure efficiency, increase almost proportionally, 7 times and 3 times here. So this was an interesting case to understand why. Why does it have more efficiency if it's less incorporated? So that is also work of Diana. One of the data that is important that we should know is that when you incorporate the photosensitizer in tyrosphere, it makes a lot more single oxygen. You see, this is the emission from both dyes when incorporated in the tyrosphere. And you see, it's basically the same yield as in methanol. In water, it's smaller. And the reason why it's smaller is because in water, these molecules aggregate. You see here is the absorption spectra and the emission spectra of these two molecules in methanol in water. Water is aggregated. And in thyrosphere, you see that absorption spectra and emission are basically the same in methanol and in thyrosphere, showing that there is stabilization of the monomer in this polymer and making a lot more single oxygen. Colocalization, well, where are these molecules located if they are delivered with the polymer compared when they are delivered by free in the solution? So basically what Diana shows is that doesn't change. And you see here is the profile of mitotracker colocalization, lysotracker, when it's free and when it's in thyrosphere. The level here is the amount of fluorescence, so it doesn't really matter, but the ratio here is basically the same, and the ratio there is basically the same. So this molecule binds more to mitochondria as the free dye, and this one binds more to the lysosome as the free dye. When we do cell death mechanism, interesting, you see here, 3 hours after damage, the molecule delivered by thyrosphere, the positively charged, it does a lot of propydium iodide showing necrosis. And this one, the cell is fine. 48 hours afterwards, what is this here? This is this is still, oh no, sorry. This is with thyrosphere, and this is the previous data I already showed. So basically it's the same. And here the same, incorporating thyrosphere, 48 hours. This you show lysosomes burning here, and the cells surviving if they are treated with the positively charged dye. The same as we saw, very similar as we saw before with the free dye. So now, for me, there is a problem here in this. I mean, we solved this in my view, but I want to put the question here. So, why are these molecules more efficient if they have the same mechanism of cell death? Considering also that the only data that we have is it generates more single oxygen when they are bound to the polymer. However, the polymer not really go to mitochondrial lysosome. It should be somehow releasing this molecule, which goes to the site of localization. So we need to prove that release, and Tayana did that by using lifetime fluorescence measurement, microscopy measurements. So this is the lifetime inside the cell of the free dye, inside the cells like this. And when we have delivering lysosomes, we also have, in this thyrosphere, we also have this decrease in the lifetime. So what we propose is happening is the following. The molecule enters as a complex, and it gets partial release here. The ones that goes to the site, they choose go to mitochondria or lysosome, depends on the charge. I'm not they choose. They go to either mitochondria or lysosome. However, part of the dye remains in the polymer, generating a lot more single oxygen. So increase the amount of damage by targeting other sites. Okay. So if delivered this dye, positively charged dye, in the thyrosphere, it goes to mitochondria, and some of them stays generating single oxygen within the cell, and for the negatively charged goes to lysosome. Again, what is the advantage of going to mitochondria or to lysosome? Because they can engage in these reactions that are contact-dependent reactions. And these ones that are remaining in the polymer, they make single oxygen. So you do both, single oxygen and contact-dependent reaction. Okay, so I think we answered this, how to improve the photosensitizers. We have to think of multifunctional, and in our view, we have to do localization and both type 1 and type 2 photochemistry. So before I finish, I was not joking. There is an open opportunity for postdoc in my lab. Here is the link. You can talk to me, send me a message. It'll be open till next week. There is more information here. The opportunity here is not related with PDT, but related to the effect of sun in the skin. The reactions are basically the same, but the photosensitizers are natural. They are in our skin. Okay, but more details we can discuss later. So this is another picture, different one, an older one actually. Tayana is still there, but I'm not wearing the tie. I was wearing the tie when I was in rectory for the international office, but usually I'm like that, a bit older now. So thank you very much.
Professor, for the very interesting talk. We have time for a few questions.
Yes. So very nice talk. And I was wondering, curious, have you using mitochondria penetrating peptide for the study?
Yeah, we have used it. I mean, we have some data. It's because the efficiency of these dyes to go to mitochondria is so much. I mean, mitochondria has 100 minus 180 millivolts negative potential. So they all go there. The penetration in the cell depends on the polymer. It may be a issue, as you mentioned. So perhaps you're talking about putting
No, I meant about these Professor Shanna Kelly in University of Toronto. I was sitting on the...
Yeah, I know him. I know
so
the...
Yeah. Mitochondria penetrating peptide, I think they stopped for a while now. It's restart the program. And I think there is some new version of the MPP, the peptide, actually showing very well not only the distribution intracellularly, but also for in vivo stability.
Okay, I'll take a look.
Look it out.
Okay.
And the second question I have is, many years ago, the PDT people always shy away from DNA, right? But many years ago, I think 20 years ago, the Russian group did a study. I still remember. They were showing like a 10 to 5 difference, 5 order of magnitude difference between the sensitizer targeting 2 mitochondria versus the targeting DNA. And I remember, Brian Wilson's now tried to restarting this process doing the DNA targeting. Have you ever think about the DNA targeting?
No, we didn't do DNA targeting. Yeah. So I also tried to target the mitochondrial DNA. It may be a practical way to improve efficiency. That probably raise a lot of issues. I mean, if you don't kill completely the cell, I mean, how can you make sure you're not really damaging it, transforming it? I don't know.
I think the study has not done enough to totally shy away from that.
I see. Okay.
I think it's time to go back to it.
Okay, let's talk about it. Thank you.
Do we have more questions?
So I was wondering when the nano formulations were formed. So first question is, do we know the size of those nano capsules that were made out of those photosensitizers? And secondly...
You mean tyrosphere?
Yes, tyrospheres. So the size of the tyrospheres, and if they are in the nano dimensions, then probably the mechanism of internalization would be endocytosis.
Yeah, it is.
So I would think that once they get internalized into the cells, and the photosensitizer just gets separated out of the tyrospheres, so there is a probability of those photosensitizer molecules to get diffused into the nucleus and kind of cause some nuclear damage. So any studies done for identifying if there was any nuclear damage done?
Yeah. I just showed one data, not with the tyrosphere, but with a normal dye, enters the cell by the same mechanism, endocytosis. I show you the DNA, the nucleus DNA was intact, and mitochondrial DNA were all damaged. The reason why these dyes do not go to nuclei is that the nuclear membrane is a very protected membrane. So it's a very controlled thing, allowing things to go in. Not many molecules go in. So there are molecules that stay in the nuclei, but positively charged dyes don't go in. Okay, so they do not damage the nuclear.
And that would be the case for the negatively charged tyrospheres as well?
Yeah, because as you mentioned, it is partially released, and I'm sure the polymer itself does not go to the nuclei also. So.
Okay, thank you.
Anyone else? So let's thank Professor Baptista again, please.
Thank you.